Hey guys, I'm going to go through the notes with you today. I hope you're all doing well and had a great weekend. So here we go. It might be a little slow, so I apologize if that is the case. But we've been talking about different chemical reactions of digestion, and to kind of understand a little more how digestion works, we're going to look at specifically at enzymes. And I'm going to be doing a lab for you guys later today or tomorrow, we'll see, where I show you guys the lab. So don't do the lab investigation part until I have shown you the actual lab, okay? Alright, so we're going to look at enzymes. And what enzymes do is they speed up chemical reactions by lowering the activation energy. And the activation energy is the energy that's required for a reaction to happen. So usually we have to put energy into a reaction to make it happen. If we want to break apart, let's say, a starch into its sugars, it's going to take energy to do that. Okay, and an enzyme makes it so that takes less energy, and I'll show you how that happens in a little bit here. So how important are they? All biological processes need enzymes to work. If we didn't have enzymes, the chemical reactions in our bodies would not be able to happen at a rate fast enough to support life. So some of those processes include making new macromolecules like proteins and building muscle and DNA and those kinds of things. Breaking down molecules, so like digestion, we also call that catalysis. Making energy for the cells, so they're involved in cell respiration. If you remember back to biology, cell respiration takes in oxygen and sugar and changes that into a chemical form of energy called ATP. And that ATP is used by the cells to do different work. Also, it helps in participating in mitosis. So remember, mitosis is making identical copies of cells to replace old cells or injured cells or things like that. So enzymes are catalysts, and catalysts, again, speed up a reaction. So they're found in living things. They speed up the process. This just shows you that we have a sugar plus oxygen, so this actually is cell respiration. Without the enzyme, it would take this much energy, compared to with the enzyme, it would take this much energy. So there's quite a difference in, from here to here, a difference in energy required without the enzyme. Okay. So these are called exergonic reactions, which means that energy is being released. Okay, and we know the reactants have a higher energy than the products in an exergonic reaction. Okay, so reactants are what we start with. Our products are what we end with in a chemical reaction. Endergonic is the opposite. We have energy being absorbed in the reaction. So the reactants have a lower energy than our products. Okay, so what we start with has a lower energy than what we end with. Okay, an enzyme is a protein, and they're specific to specific reactions. Okay, we have to have a specific enzyme that breaks down starch. We have to have a specific enzyme that breaks down proteins. We have a specific enzyme that breaks down sugars. They're specific to each reaction. And they each have a specific shape. And we've talked about shapes a little bit when we did protein, and we talked about denaturing. When we denature a protein, it changes the shape, which changes what the protein is able to do. So if we have a certain enzyme, let's say, that breaks down starch, and it's denatured, it won't be able to break down starch anymore. Usually enzymes, we know they're enzymes because they end in ACE, A-S-E. Okay. So sucrase breaks down sucrose. Proteases break down proteins. We talked about salivary amylase breaks down starch. Okay? Lipases break down lipids. DNA polymerase builds DNA. Those are all ending in ase. Another important thing about enzymes is they're not used up during a reaction. Okay, so let's say we call this a substrate, so this is what we want to either, it's basically the reactants of our product, or our reactants in our reaction. Okay, they have a specific shape, and they fit into the enzyme in a certain spot called the active site. Okay, and you'll notice it's like a puzzle piece. The shapes fit. Okay, so once they bond, once the reactant or the substrate bonds to the enzyme slightly will change shape in some way, either to bring the reactants close together or to pull them apart. In this case, it's going to pull them apart. So it breaks these two apart, and then they leave. Okay. And then when that happens, the enzyme shifts back to its original shape so that another substrate can come in and do the same thing. Okay. Their enzymes aren't changed by the reaction, used temporarily, and they can be reused again and again. So we need very little enzyme to help in these reactions. I don't know why this is like this. Okay. Again, it's a lock and key model. Okay, a specific shape fits in there. So the shape of the protein allows that substrate to fit into that active site of the enzyme. And again, there's a specific enzyme for each specific reaction. Some vocabulary. We have an enzyme, which is our helper protein. It speeds up a reaction. The substrate is the molecule the enzymes work on, the reactant in the reaction. Okay, the products are what's being produced at the end of the reaction. Okay, so reactant, product, and the active site is where that substrate attaches to in the enzyme. So what affects enzyme action? It has to have the correct protein structure, which means that it has to have the right order of amino acids. Remember, your proteins are made up of amino acids. They have to have a specific shape, especially in that active site, because that is where they're going to be bonding to. Usually there's a certain temperature. There's usually a certain pH where those enzymes work the best. If we change, I'm going to go back, if we change any of these things, it can affect how this enzyme works. It can slow it down. So our optimum temperature for humans is 35 to 40 degrees Celsius. So basically body temperature, which makes sense, right? We want our enzymes to work optimally at our body temperature. High temperatures are greater than 60 degrees Celsius. What's going to happen again is that the protein is denatured, it loses its shape. At lower temperatures, the molecules just move slower, so the substrates are going to react less with the enzyme. So high temperatures and low temperatures are going to affect. How does pH affect it? Well, the change in pH can also change the protein shape. Most human enzymes operate between 6 and 8 on the pH scale, which is it's neutral. 6 is a little acidic, 8 is a little bit basic. Depending on the enzyme, depends on what our optimal pH is. For pepsin in the stomach, and we're going to talk about this one next week, I had to order some materials. This is in the stomach. So pepsin is an enzyme, but it requires a pH of 3 to work well, which is very acidic. It makes sense. You have acid in your stomach, right? Trypsin, which is in your small intestines, needs a pH of 8, so slightly basic. So in summary, we've got enzymes are essential for biological processes. They're totally dependent on our protein structure. They're affected by changes in temperature and pH. They convert substrates to products or reactants into products. I can't read that one. And they are reusable. Alright, so once you have watched this, if you guys go into classwork, your worksheet here just goes through kind of what we just said. You won't be able to do this part where you draw it, right? So you can skip number 5, but you'll do the rest of these questions here. Okay? Let me know if you have any questions. Again, don't do the lab until I post the video about the lab. So hopefully I'll be doing that today or tomorrow. Alright, have a great day. I miss you guys.